Morning, students. This is for the 5th semester students of B Farm. We are just continuing with the chromatography class. I would like to continue with TLC, means thin layer chromatography. Thin layer chromatography is a very interesting and important experiment, and also a very important technique which is used for isolation of compound, identification, even qualitative and quantitative test. Usually qualitative test we are combined with even column chromatography, which we studied in the last class. We used to confirm the compound by means of using thin layer chromatography. So we should know about the basic principle behind, and as well as thin layer chromatography, even advanced techniques have been made now. High performance thin layer, that is HPTLC, thin layer chromatography, high performance thin layer chromatography is now every pharmaceutical industries have been used for a very accurate and fine results for the isolation and identification of compound, as well as quantitative and qualitative. TLC is an example of adsorption chromatography. It's an example of adsorption chromatography. But according to the thin layer which you are keeping as a stationary phase, even it can be changed to other principles also. That also I have made a discussion for you. But usually we can say it's an example for adsorption chromatography. The stationary phase being, we are talking about the stationary phase. If you change the stationary phase, it may change the principle, but stationary phase being the thin layer is an adsorbent held on suitable backing. Separation of compound present in plant extract depend on the difference in their adsorptive affinity, absorptive and desorptive removing behavior in respect to the stationary phase. So that's the very important point which we should note it down. Thin layer chromatography, TLC, involves a thin layer adsorbent mixed with the binder such as calcium sulfate, that is gypsum, which is always, we used to say silica gel. G means, G stands for gypsum. Calcium sulfate is a binder because it has to make a thin layer in a glass plate or a thin holding plate. It should need, or it has to have a binding agent. It is already mixed with calcium sulfate. So which is spread on a glass plate, allowed to dry. Even we can use the word after drying, even go for activation also. The plant mixture to be separated is applied as a spot near the base of the plate. This is the practical events. When I was discussing in the class, I was telling about how we perform a thin layer chromatography in the lab by means of using a chamber as a beaker. Can also be followed, which I explained you, that will be easy for you to remember and to write the theoretical knowledge. The practical knowledge can be easily expressed to write in the theory. The plant mixture to be separated should be kept as a spot or a layer of spot near the base of the plate, which is then placed in a closed glass tank containing a layer of developing solvent. So that is, the mobile solvent is developing. In the picture itself you might have shown there is a 45 degrees inclined. You are keeping the thin layer plate and the chamber, and the solvent which is kept in the bottom is the mobile solvent phase, which is shown there. You can see this is more visibly. We can see the solvent and the spot which is kept in the as a sample, and we have shown as a picture which is getting separated, and you are getting a green color, red color and an orange color, which you kept as a sample as a dark color, which is mixture of compounds, can be get separated. In this means when you keep 3 spots as shown in the picture, it is possible for you to get separated by means of running against the gravitational force in the thin layer which is framed in the glass plate of thin layer chromatography. So this is the original chamber for a thin layer plate of TLC. You can see the plates are very large and square type and rectangular type of chamber, which is large one, which we kept as a fluid which is kept at the bottom, which we can see in the picture is the mobile solvent. So we have discussed about the baseline, and also we have discussed about the sample spot which should be kept above the level of the solvent system which you kept in the chamber. That is very important. We have discussed in the class. I have told you very clearly, when you keep the spot below the level of the solvent system, the spot may be dissolved in the mobile solvent and you may not get the separation properly. So you have to select the baseline where you have to keep the spot of the sample should be above the level of the mobile solvent present in the developing chamber. This is called the developing chamber. It can be even used as a beaker, can also be. The beakless beaker will be very nice for us to keep in the laboratory. It is possible even saturation can be done by means of keeping a filter paper in the sides of the beaker. Saturation of the mobile solvent towards the chamber is very, very important for you to get the movement of the spot and the separation of the spot, and also the movement of the spot and the development of the separation of the chromatogram is very much important for the saturation of the solvent system in the chamber. That is also we have discussed in the class, such that why we need the saturation of the vapor in the chamber, because when you get a chamber, the movement of the solvent system through the stationary thin layer will be uniform, and also you will not get, you can avoid from the edging effect, and as well as the effect of the RF value which you calculating will be perfect. So this is what some of the problems can be solved, and also which we have discussed. The solvent moves up the plate by the capillary action, carrying with the less strongly, carrying with it's the less adsorbed component of the mixture very faster, because one which is having less affinity, one which is having less strongly adsorbed in the stationary phase will move faster with the solvent system towards in the top. Therefore the RF value will be more, and also it will be move faster and get separated from the mixture of the compound very faster. What we have discussed in the column chromatography, the same system will be followed. The affinity, if the affinity of the compound in the present in the mixture is less, then the movement of the compound in the stationary thin layer will be less. It will move slowly. If the affinity is less, it will move faster. Sorry, I made a mistake. And if the affinity of the compound is more towards the stationary phase, it will stay for a longer period. And also you should know that if it is an adsorption chromatography, if you are having a solid stationary phase in the thin layer, and in the TLC, it works in the adsorption chromatography. So the adsorbent which you made in the thin layer, maybe silica gel, maybe alumina, maybe cellulose, if it is kept as in a silica gel or alumina, usually we are having the thin layer, they are solid in nature, and also the mobile phase will be the solvent, so that it will work in the adsorption chromatography. So the adsorbent capacity of the sample which you kept as a spot plays a major role in the separation of the compound. While that's why this line is written, while the more strongly adsorbed compound remain near to the base of the plate, because its movement are getting separated from the mixture, moving through the stationary thin layer will be slow, because it has strongly adsorbed in the stationary phase. When the solvent reaches to the 1 to 2 centimeter from the top of the plate, or 3/4 of the plate, because we should not run up to the plate end. That is what we are mentioning, that, and the plate should be removed from the tank and should be dried. Now the separated compounds of the mixture appear as the spot which is shown in the picture on the finished plate. If the chromatogram, if the compounds are colored one, if it is colorless, then we have to go for detecting, or we have to go for detecting liquids or detecting reagents to be sprayed, or by means of UV spectrophotometric observation, or with the fluorescent observation, if the compounds are having fluorescent effect, then we have to change the nanometer of the UV light to be fall on the plate, can be identified the spot. It's also possible, corresponding to the bands of the adsorbent column. And what is with the adsorption TLC, different substances have different adsorptive capacity. Any one material can be vary its activity according to the pre-treatment of the TLC plate. The adsorbent must be chosen in relation to the property of the solvent and the mixture to be separated. What type of compound you are interested to get separated, what is the chemical nature of the compound, these things will make to select the relation property, the select of the solvent system, which are the adsorbent which you are selecting. In general, if a highly active adsorbent is used, then a solvent with a corresponding high power of elution from this substance will be required. Example, aluminium. It has different categories of acidic, neutral or basic. With a different activity, grades is commonly used. To produce a film with reasonable handling properties, the adsorbent may be mixed with 12% of binding agent like calcium sulfate, which we have discussed, gypsum, to act as a binder. So the student should know what is the percentage of the binder needed for any type of solid material to be made in a thin layer plate for TLC. Ready mixed powder are commercially available. Already they have made, gypsum is added, silica gel G is available for TLC plate which we can prepare. They needed to be mixed with a certain amount of water, may be spread onto the plate with a glass rod. The film sets within a few minutes, is then activated. Activated, you just note it down, the students, activated. The unwanted, because you just made in the previous paragraph, the powder is commercially available, already mixed with the gypsum, and we use a solvent system to make it as a slurry, and we are spreading it with a rod to the glass plate, or dipping method or spraying method is also possible to make a thin film of thin layer chromatography. But we are using some solvent for them to make it as a slurry, so that solvent should be removed, and the water which may be present or the solvent which may be present may be hinder your adsorption capacity of the separation of the compound. So for that, activation is needed by heating at a suitable temperature. Usually 110 degrees centigrade or 115 degrees centigrade for 15 minutes will be more than sufficient for make the thin layer to be activated, or make the adsorbent to be activated, or to make the adsorbent or the stationary phase to be ready for the preparation of chromatogram, are to ready for the separation of the compound by means of its adsorption capacity. So solution to be examined or applied to, that is a sample to be examined, is applied to the film or a spot with a help of capillary tube or by means of microsyringe. If you want to go for a quantitative estimation, you should know what is the amount which you have kept as a spot. So microsyringe can be used, or micro pipettes can also, micrometer pipettes can also be used, or capillary tubes are also can be used for this purpose. So TLC solvents, I have added some solvents, but usually before in the last class also I told the student should know from the starting from the low polar to the high polar what are the solvent system. There are many, but still I am telling some few numbers which I told in the last class also, like starting from petroleum ether, hexane, benzene, carbon tetrachloride, chloroform, acetone, methyl acetate, ethanol, methanol, dilute acids or water can be increasing polarity. Like this solvent system you should know. There are many other solvents are also commonly used solvent, methanol, ethanol, and other alcohols, chloroform, ether, ethyl acetate, acetone, methanol. There are many other solvents are also commonly used for the separation of the compound. Visible of the spot, I told sometimes colored compound can be seen, but otherwise you have to go for detecting reagents. Like for ninhydrin can be used for amino acid, drag and drop reagent can be used for alkaloids, and anisole solutions can be used for glycosides, and also spotting can be done by means of visible or UV lights, or fluorescent compounds can be seen in with different nanometers like 360 nanometer light or 254 nanometers, which can be easily spot can be identified, which we have UV chambers are available to see the spot clearly and to calculate the RF value for the qualitative estimation and quantitative estimations can be done in TLC. If visible spots made visible by heating a specific period of time, examine under UV light if the substance are fluorescent, and spraying the finished chromatogram with a suitable reagent, or example iodine drag and draft reagent are used for spraying general detection of iodine, which is specifically for alkaloids, which you can see that the purple color and the red color spots which you have seen by means of using drag and draft, and also yellowish brownish spots can be seen when you are using iodine chambers or iodine vapors which is created in the chamber. Advantages of TLC over paper chromatography. Separation of compound can be achieved more rapidly with less plant materials. Separated spots are more compact and clearly demarcated from one another or each other. Reagents like as concentrated sulfuric acid would destroy a paper chromatography, but which can be avoided in the TLC. So that's an advantage, but may be used in the locating separate substances on the TLC plate very easily when compared to paper chromatography. Simple and inexpensive, quick results can be achieved between 30 to 40 minutes or 1 hour maximum. Good separation of spot compounds can be seen, very sensitive. The chromatogram is resistant to the action of chemicals used for the visualization of the compound, which you are spraying like iodine, sulfuric acid, or you are using drag and draft reagent, Hager's reagents, or even any other reagents like anise solutions, whichever you are using. All of them can be having a resistance because it is made up of glass plate. Components of TLC, just we can make it is a revision which we have discussed in the class, also in the practicals. You can see there is one is adsorbent mainly, which selection, and eluent, that is mobile solvent which you are selecting. Then the substance to be separated, what type of compound you are going to separate, what type of alkaloids or glycoside, what is the need, what is the type of chemical you are interested to separate is very important. Not only for herbal formulation, this TLC is very much useful for even the synthetic products, or even in the pharmaceutics of them, or in the F&D formulation and development technology, and also formulating all the analysis and quality control and quality assurance also plays a major role for this TLC technique, or even thin layer chromatography by means of high performance thin layer chromatography. HPTLC is advanced technology now. It is more useful in the pharmaceutical industry. And this is a plate just to show you how the eluent is kept and the distance travelled by the spot and distance travelled by the sample solvent front for RF value which we calculate. The adsorption capacity or adsorptive capacity of the adsorbent are related to their polarity. This type of points is very, very important. The more polar the adsorbent material, the better will be separated compounds like aluminium and silica gel can be used in this case. Under normal conditions, these adsorbent hold little water. Adsorbed organic material, these decreases the adsorptive capacity of the material. Heating may activate adsorbent. The eluent, that means the mobile solvent, the more polar the solvent, the better is eluting, desorption, removing from the stationary phase property. Therefore, water is better eluent than ether, which in turn better than cyclohexane because it is low polar solvent hexane. Polarity also plays an important role. The least polar substance or components Example, saturated hydrocarbons are least strongly adsorbed, while the highly polar compounds like example acids are strongly adsorbed. So these type of examples, if in practicals or in practice only two adsorbents are widely used, the silica gel and aluminium. When used these adsorbent, the rules is to match the polarity of the developing solvents with that of the compounds of the mixture to be chromatographed or to be separated. So RF value, and also I have asked the question why we need the RF value. RF value is the distance traveled by the substance from the reference line or the baseline in centimeter divided by the distance traveled by the solvent friend from reference line centimeter. That is, you should calculate by centimeter. Usually the RF value will be below 1 because the denominator will be always high because it is taken as the solvent friend, the maximum solvent which you are used. Usually sample will be moving less than the solvent friend. So this is about the picture which I have taken from the biojuicer. That is same thing, same. They have used a beaker actually. But the chamber for the TLC plate it's a rectangular tank. It's a big TLC plate. You can see in your laboratory. In chromatography, RF value are the most basic prerequisite of the experiment. These numbers indicate whether the analyte or the solute or the sample which is separated prefers the stationary phase or the mobile phase. Which the stationary phase and the mobile phases are with the stationary phase and the mobile phases the separation takes place. RF value are used to determine the polarity, relative masses and relative solubility among other things. Additional factors, separation of alkaloids, what all things you should remember. I have written additional factors before that. Alkaloids, they are nitrogen containing, basic in nature. Moderately polar solvents are good separation, like you can use ether and ethanol in the ratio of 95:4. The more polar solvents, example pure methanol when they are using for alkaloids, would be preferably absorbed. The alkaloids would be carried along with the passage of the solvent. So when you are using an isocratic solvent like methanol for some mobile phase, what happens? The separation was completely disturbed because it was very much soluble in methanol. The alkaloids have been taken very strongly towards the end of the line where the solvent friend goes. So the RF value determination will be very difficult. So the resulting is a poor separation because of high polarity of the methanol. On the other hand, if the non-polar solvent like cyclohexane, if you are using for separation of the alkaloids, what happens? The spot may not move, would be unable to displace. It cannot move the sample which you kept in the baseline. The alkaloid may be adsorbed layer, it will stay in the same place and it will remain on the near of the baseline. So there is also no separation. So what is the additional factors which you have to remember? See, for example, first line itself, you have to remember, you are adding ether and ethanol in the percentage of 95:1 is the correct polarity of the mobile solvent which can use for the separation of alkaloid. This is crude way which I told, but still the polarity of the solvent have to be decided by the researcher by trial and error method he has to identify. Additional factors to separation of alkaloids which have to be determined is if using an aluminium thin layer or aluminium as a adsorbent layer, if you are using, is a neutral. A neutral solvent should be used. If you are using silica, used it as slightly acidic due to method of preparation. And the alkaline solvents such as acetone, water, 25% ammonia will be useful. 90:7:3 for the good separation. This is what we are just giving us some example. The solvent systems can be changed by means of increasing, decreasing the polarity according to the compound which you are interested to separate. Separation of volatile oil, again I have given one example. When you want to separate volatile oil, the constituents of volatile oil are mainly non-polar. Terpenes, you know that volatile oils are made up of terpenes. They are volatile in nature. They are the basic components which is prepared from the terpenes like isoprene units. are therefore best separated with the corresponding non-polar solvents such as chloroform, benzene mixtures. So you see here we are using chloroform and benzene mixtures are very good for separation of volatile oil principle as they are terpenes in nature. Certain oils may be in more polar solvents like cloud, which contain eugenol and phenolic compounds, can also be used more polar solvents, little more polar, not like chloroform, benzene. You can go for methyl acetate or even little more ethanol or methanol can be used. Separation of sugars and the sugar acid mixtures. Sugars, carbohydrates. So these mixtures are produced hydrolyzing starch, gums. You will get carbohydrates. Polysaccharides can be made into monosaccharide, disaccharide or trisaccharides. You can get. They are strongly polar, are soluble in water, strongly polar, are therefore strongly adsorbed in the silica and alumina layer. Although strongly polar solvents are used, separation of these thin layer is not generally satisfactory. Better results is obtained using weak polar adsorbents such as cellulose. So they can be preferred for paper chromatography usually for carbohydrate isolation. Cellulose. And even cellulose can be made a layer in the thin layer of plates, and also you can go for separation by cellulose using even thin layer chromatography. Polar solvents such as butanol, ethanol, water in the ratio of 5:4:1 can be used alternatively. Paper chromatography is the best technique which can be preferred for this carbohydrates or you can see sugars. TLC is comparatively better separation tool than paper chromatography. Though the adsorbent term use various types of TLC, but principle of separation may be any one of the four fundamental mechanisms of separation can be followed. Ion exchange chromatography can be followed, partition can be followed, adsorption can be followed, molecular exclusion can also be followed by means of changing the stationary phase. So you just see how it is possible in the thin layer chromatography. Principle of separation totally depends upon the coating substances made on the plate. What type of coating substances you are applied. From the beginning I was telling you. Understand? So if you are using silica or alumina as applied as a thin layer coating substances, adsorbent which is used in the thin layer chromatography, it works in adsorption principle and it takes place. Okay, it is a solid. So we should remember that what type of adsorbent you are using. But if you are coating with Sephardex, molecular exclusion will be the principle. Sephardex, matrix, Sephardex, proteinous nature, Sephardex, gel. When you are having a thin layer of gel, Sephardex, if you are having a coating in a glass plate, then it will work in molecular exclusion principle of separation, that same TLC. And the same another TLC, if you are using ion exchange, resins will be, if you are making as a coating mechanism of separation, if the resin is coated as a substance in the thin glass plate, then it will work in ion exchange chromatography. If you are using cellulose, a coating, immediately it will work as a partition chromatography. Then the student will ask, cellulose, how it is possible partition? Partition chromatography means the stationary phase should be liquid. Cellulose is a solid. How can it be a liquid as a stationary phase? Cellulose always surrounded by water molecule. That you should know. Even paper, we cannot see the water molecule, but all it is made, paper is made up of cellulose, and all the cellulose molecule is surrounded by water molecules. That water molecule will act as a stationary phase, and the mobile phase which you are using also liquid. So two liquids, two immiscible liquids, the water is aqueous and you are using a non-aqueous solvent as a mobile phase, then automatically there will be an immiscibility, that is will work in the partition chromatography. So thin layer chromatography also, it is possible for you to go for partition chromatography if you are coating the glass plate using cellulose. So most often the adsorbent are applied on the glass plate. The dimension of the glass plate may be vary from 20 into 20 centimeter to 20 into 5 centimeter about the dimension. Maybe square or maybe rectangular. Sometimes even microscope plates which you are using, the small slide, can also be used for your single component separation, for your quality control test. It can be used even in the laboratory. I will show you when you come to the laboratory. Even the micro slides can also be used to examine the process of chemical reaction of thin layer chromatography method. The glasses are coated with adsorbent in different techniques like pouring technique, dipping technique. Slurry is made and dip the slides and you can take a thin layer. Spraying also can be done. Spreading technique by glass rod can also be done. And also there is an instrument which will be having a spreader is also available in the glass plate of TLC plate. Allow them for drying, and theoretically the methods used students how to read and understand the method of preparing, how they are doing pouring, how they are doing dipping method for thin layer of glass plate, thin layer of aluminium, thin layer of silica, how we can make. This is the method of preparation of thin layer chromatography is also needed for the students to know. In all the chemistry papers they will ask about how will you prepare a thin layer chromatography and how will you make, how will you prepare, how will you perform a thin layer chromatography is very important for the students should know before in all degree level of pharmacy students. Allow them to air dry and activate them in 100 to 120 degrees centigrade for an hour. Activation is very important. As I told, unwanted slurry preparation, when you are using the water, that water will hinder for your experiment which you are performing according to the stationary phase. When it is solid means it should work under adsorption. Chromatography means it should have the capacity to absorb the compound and to make the separation. So there should not be presence of unwanted water which is used for the purpose of making thin layer should be removed. So 110 or 100 degrees centigrade for at least 1 hour should be kept in the hot air oven for getting activation of the thin layer chromatography which is prepared by us. And the samples can be applied by micropipette or micro capillary tube. Micropipettes can also be used because suppose if you want to go for quantitative estimation, it is needed how much ml I have kept as a sample. So it should be spotted at least 2 centimeter above the base or on the such height, and the spot area should be immersed, and the spot area should not be immersed into the mobile phase. This point also I have explained to you, very important. The baseline should be above the level of the solvent system, level of the mobile solvent system which is kept in the developing chamber, because once it is touching the baseline or once it is touching the spot which you kept, it will be solubilized in the base of the solvent system and it may not get movement towards against the gravitational force, and to get the separator in the stationary phase will be disturbed and it will be changed. So it is the area of the spot sample should be minimum with sufficient concentration. That is very important. Mobile phase and the thin layer chromatography plates should be kept in the developing chamber with a large or a beaker. The chamber should be saturated. I was telling about this saturation. Which mobile phase? Otherwise, edging effect may happen. What do you mean by edging effect? Curve. No straight line of the solvent system movements may be there. So these all can be avoided when you are having edging effect can be found. Which is the solvent friend will be in the middle will be faster and the side will be slow. We can see the picture I have shown in the next slide. The mobile phase selected on the basis of phytoconstituents which has to separate and the nature of the stationary phase. It may be a single or a mixture of solvent. Single means you may say isocratic, mixture of solvent we will say gradient method, and the polarity of the solvent adjusted such that it easily separate the phytoconstituents. You see, this is the picture which I am showing. The right side picture, it shows a straight line of the solvent front. Left side picture, you can see a curve. This is called edging effect. The center part of the solvent system moving against the gravity will be faster if you are not saturating the chamber. You can see the saturation chamber, you can see the two pictures. The side of the right side picture which is below me, there is a blue color line, that is the filter paper which is kept. Now one side if you keep also it is sufficient, but they are shown in the picture very clearly that is a filter paper which is kept. So the filter paper will take the solvent system slightly to the above, and so that it will saturate the chamber. That is an arrow which you have showed. The saturation will take place so that you will not get this type of edging effect can be avoided. So you can see the chamber With normal saturation, the arrow stands for the evaporation of the elute, and the layer dots and symbolize the vapor density. So you can see, if suppose you are not having saturation, there will be vapors can also be evaporated, so that edging effect will take place. In the center part, it will be movement will be very faster than when you see the edging effect. So the chamber line, the filter paper, blue color which is shown, that is a filter paper, saturation of the eluent vapor is shown in the picture. The different development techniques used in the TLC are one-dimensional, two-dimensional, horizontal and multiple development techniques are also there. One-dimensional, suppose if you are to in this one position x-axis, and the same plate after taking another 90 degrees can also be two-dimensional development can be done for getting more separation. In one-dimensional vertical technique, the mobile phase flows against the gravity in the capillary tube. Most separation is done by this technique, in which one spot is applied at the corner of the plate and the plate is developed in one axis, dried it. Further, run the plate in 90 degrees and develop the plate one another axis. After development of the chromatogram, the isolated compound can be visualized by detecting agent. Detecting agent can be two types. One is destructive type, another is non-destructive type. Destruction. So there is no reversible, understand? Irreversible. So you can say in destructive type, the sample cannot be recovered. You cannot take the sample. It's only qualitative estimation, only identification, or it will be destroyed due to the chemical reaction of the spray reagent which you are spraying on the sample. While non-destructive, the sample will not get spoiled. You can take the sample back and you can analyze, and the quantity of the compound can also be identified, and you can go for other tests also. The non-destructive method, sample can be recovered, that is the difference. And non-destructive method, sample can be detected by UV chamber method, or densitometric method, or even iodine chamber method can be used. For quantitative purpose, the density of the spot can be measured, or the spot can be eluted with the solvent and analyzed by conventional technique like spectrophotometric method and electrochemical methods can be used. Now we want to discuss about the paper chromatography. So I think we will continue in the next class. Thank you.